And we'll start the note. And you can write down the main title here, unit, and this is actually unit 3. We don't put a number on it because sometimes we change the order depending on how many teachers are using the equipment. And this is on tissues, organs, and systems of living things. And that's exactly the order in which we do this. First, we learn about the cell and about the properties of the cell and the different parts of the cell. We then talk about how cells divide and how they can reproduce, that type of thing. We then talk about different types of tissues in which the cells can be representative. And then after that, we talk about how the different tissues can come together to form larger structures like organs. And then we talk about how those organs can work together to produce an entire system, like or a living thing like a human being or a cat or a plant or something like that. So it's really just the breakdown of the biological systems, starting from the simplest, working our way up to the complex, which is, I guess, the way we kind of teach the chemistry as well. We talk to you about the simplest part of chemicals, the atoms, and we work our way up to more complex things. The difference between chemistry and biology is that I know chemistry really well, and I don't know biology very well. So I'm hopefully not going to trip over too much of this stuff. But this is going to be more note-dependent and less practice and, doing that type of thing, and more information-dependent. All right, so hopefully you can get information out of the notes. And you can ask me. I can always go to someone who has more biology knowledge than myself. Mrs. Mayhead and Mr. Edgar are our resident biologists, and so I could always get some information from them if we need it. Okay, so we are going to write this stuff down. These are just the basics. And I'm going to bring the whole thing up. Oops, back up here. Now, much like the chemistry, I'm pretty sure this information is not all new to you. In terms of the optics, it's probably fairly new. Probably not ever studied light in that way. In terms of biology, I'm pretty sure, like I said, I can't remember if it's grade 7 or 8. All I know is when my son got to this unit in the grade 10, he was losing his mind because he said, we did this all in grade 7 or 8. He was complaining. So it is very similar to that unit you did back then, but elementary schools are different, right? Different teachers who have different comfort levels with different subjects. You might have talked about it in detail. You maybe didn't talk about it in very much detail. Yep. As soon as I get Shane to write the test. So he's the only one left. So I'm going to assume he's going to come tomorrow. And if he's here tomorrow, then I can give it back to you at the end of class, that type of thing. Marks are already posted online. So you can see what you got in all the sections by looking at that. But so marks are already posted online for those who wrote it. Now, this little quick discussion of the cell and this cell theory, I actually do have a little bit of background knowledge on it because the scientist that is kind of credited with kind of starting this idea, his name was Robert Hooke. He lived about the time of Sir Isaac Newton, so just before Lavoisier, the chemistry guy, and Newton, right, he's the physics guy. Robert Hooke was actually a physics guy as well. If you take physics with Mr. Petrus, you'll learn something called Hooke's law, and that governs the way springs and elastic objects behave. Robert Hooke was a fairly smart guy. He was a very bitter man. he felt that he had came up with the laws of motion before Sir Isaac Newton did, and basically had a little hissy fit stating that, no, he came up with the rules, but there's no information to support that. But anyway, Robert Hooke was quite a diverse scientist, and you could be back then. You could dabble a little bit in chemistry and dabble a little bit in physics and a little bit in biology, and that's because everything was so new. No one had really written down any laws, so you could sit down and try a little bit, and you would have been discovering things. What Robert Hooke did was he used a microscope, and he didn't invent the microscope, but he used lenses, right? Someone who discovered the microscope. He used that microscope to look at all kinds of different structures. He looked at cork underneath the structure, underneath the microscope, and he looked at all kinds of different stuff under the microscope. And what he discovered was that there were these tiny little units, these tiny little subunits that everything was made up of. Now, whether he came up with the term cell, I don't know. I don't know a lot of the background of this, but he did write a book. It was called Micrographia, and it had all these pictures that he would have had to draw by hand, which we don't want to do because we have technology, and had all these interesting things that people didn't realize existed, and from that evolved the cell theory. Did Mr. Hooke state the cell theory? Again, that's something I should know, but I don't know, because again, it's a little more biology. But I do know that he's the one who had the first kind of looks and pictures of these things. So what did they find? They found that all living things were made up of cells. All living things are made up of cells and their products. The products of the cells. Yes, that makes good sense. All right. The cell is the simplest unit of life, and all cells come from other cells, not non-living matter. In actual fact, at one point in time, some scientists thought that life spontaneously generated from non-living matter. But again, this isn't a great story because the idea comes from living matter. Let me tell you this story. It was well known that if you put out like a piece of meat, like we're talking 100 years ago, it would work now too. You put out a piece of meat long enough, and eventually it's going to get really disgusting, and ultimately the meat is going to start to produce larvae. All right. In other words, flies are going to grow off of the meat. What are we checking here, Marissa? Checking what? Oh, you're checking marks. That's what you're doing. You couldn't handle it. As soon as I said there were marks, you checked it out. That's exciting. Okay. So ultimately, these larvae would form, and then ultimately the larvae would turn into flies, and then the flies would fly away. And some scientists thought that these larvae were just spontaneously forming out of the dead meat, right? And so they thought that life was this spontaneous generation out of something that wasn't alive anymore. Little did they know, when they weren't looking, a fly was coming along, laying some eggs in the decaying meat, and then leaving, and then the larvae came from the fly, right? They weren't watching that closely. They didn't notice that. They would look every once in a while, and they'd see this spontaneous generation. Now we know, obviously, no spontaneous generation of life. Those cells have to come from pre-existing cells. All right? Okay, that's exciting. So let's talk about the different types of cells that we can have. This is just a little bit of a flow chart, and it doesn't have to look quite as fancy as this, and I'm not a fan of the animations, but I do want you to write the title, Prokaryote versus Eukaryote, and then I do want you to write down this little flow chart here. It doesn't have to necessarily have those particularly shaped brackets there to represent it. It's just a bunch of arrows. Essentially, we're saying if you have all organisms, then those organisms can be broken down into two main cell types. And it's called prokaryote and eukaryote. And again, if I knew more about biology, I'd be able to tell you what those different prefixes mean. Pro often means before, comes before. The EU, I'm not sure what that is. Karyote has something to do with the cell. I don't have the background on that. I don't know. In a moment, there are definitions for it to explain the differences. It states here that there is a main difference between the prokaryotes and the eukaryotes. Size is the main difference. The eukaryotes can be 10 to the 5 times larger. So the eukaryotes tend to be larger, the prokaryotes tend to be smaller. So there is a size difference. There are other obvious differences, and I'm going to make you write them down in a moment. Now, you don't have to necessarily write the examples. If you did, that's great. If you drew the whale, awesome. That's nice. I know nobody drew the whale. Anyway. Examples of prokaryote, E. coli, which is a bacteria, and then single-celled organisms, which is eukaryote, an amoeba, which is a single-cell little thing there. And then we can obviously have multicellular organisms, animals, and then plants. All right, and then they also contain these eukaryotic cells. All right. Now, I think the difference is one of them has a nuclear membrane or something, or we'll find it out on the next page. My next page has all that information. Okay. So you can put the title prokaryote because now we're defining that one side. And then on the next slide, I've got the eukaryotic cells and the defining parts of that. Now, in all of the notes up to this point, I had a hand of helping what to put what order in, and I worked together because I had that knowledge. To be honest, I have had no input on the creation of these notes because that's not my strength. The teacher who put these notes together likes to put these web addresses for various kind of either TED Talk videos or perhaps some kind of YouTube stuff, that type of thing. And this one talks about how bacteria communicate. I'm not going to look at it, but my notes, as you know, are online. You could open that up. You could cut and paste that into your browser, and if you wanted to, you could get more information. But the information that's here is also sufficient. So they're the simplest cells. They lack a nucleus. They don't have a nucleus, and they also lack other advanced cell parts. Single-celled bacteria are considered to be prokaryotic. All right? I think, somebody told me, or I read this before, these, in terms of age of origin, prokaryotic cells are older than eukaryotic cells. You could take that to your desk if you want, right now, if you want to do that. Okay. So I think in terms of age in which one was formed versus the other, prokaryotic were older. In other words, they're a simpler form of life than the eukaryotic cells, which then developed more complex structures. As soon as you guys are done, you can just bring your stuff over here and I'll put it on the desk and you can take some notes. Thanks. Thank you. So eukaryote, more complex cells can exist as single-celled organisms or multicellular organisms, so they're more complex. These more advanced cells, called eukaryotes, have complex internal organization, including a nucleus and many cell parts. So that's going to be our focus for the next, probably week and a half, looking at the eukaryotic cells, looking at those structures, looking at how they divide, how they live. Again, all those parts that go along with biology and life. Protists, fungi, plants, animals have eukaryotic cells. As excited as I can get about this stuff. Sorry, I know it transfers onto you guys. The light stuff I could get excited about, but I'll pretend to be excited. It's my job. Okay. Oh, I can't do it. I can't do it. Okay. Don't write that down. So basically, we have the scenario where we have two types of main cells, prokaryotes, very simple. We're not worried about those right now. It's important. I do want you to know that they're less advanced and they don't have a nucleus and that type of thing. But it's the prokaryotes or the eukaryotes that we want to look at. So how do we look at these things? We look at them under the microscope. So that's what we want to be able to do. These cells here, anybody know what these cells are? Yep. Yeah, these are red blood cells. This guy, yeah, I think I might know what this one is. They're not animal cells. I think they're plant cells. We're going to be looking at some onion cells. I think it might be that. I'm not sure what those guys are. But again, you can see that they've got that central part, that nucleus, and that's what we want to look at, that type of stuff. Okay, so just put here the microscope as a title. Just put the microscope. Yep. The nucleus, the center. We'll talk about these parts later on. Yep, we're going to go through all that later on. In another couple of days, after we've done some microscope work, we will look at cells, and then we will talk about those structures and those parts of the cells. Thank you. Okay, so the microscope. And yes, I do want you to write it all down. Don't worry about the fluorescent dyes. And again, I didn't make this, but this blue is really hard to see. It simply says the light microscope, and that's what we're going to be using. And I was going to have you try to label a microscope diagram, but those of you who tried to print it are indicating to me that the microscope didn't come through on the handout. Did yours come through? Yours worked? Excellent. All right. Well, what was happening, it was funny because when I edit a document, I can see if other people are looking at it, and I was editing this document probably just before the end of period 2, and I could see that someone was accessing it, so maybe because I was editing the document, it was locking out that particular picture. Anyway, we'll get by. We'll figure it out. Yep. This one here, light microscope. It's a terrible, terrible choice for a font color. This is a light microscope. That's what we will be using. That uses visible light to look at things. It says there the electron microscope is 3D, but a light microscope can be 3D too. We have an advanced form of this. This is a student microscope, but we have an advanced form that has 2 eyepieces that allow you to take a look at an image 3-dimensionally. You can see that, but when you only have 1 eyepiece, you can only see it two-dimensionally. It appears as a flat image. And now we get to talk about some optics, which I like. Same with the microscopes. That's all based on science and physics. Well, not that biology isn't science. Better watch what I'm saying. I'm recording this. Biology is science. It's awesome. Before all the biologists hear this and I end up in big trouble. Okay, at this point I want to talk a little bit about microscopes and the way they work, because it's kind of handy to understand what's happening. Yeah, let's put that there. So it says there are different kinds of microscopes. The light microscope. This is a light microscope here because it uses visible light to illuminate an object, and then have that illuminated object's light come through the optics so that we can see the object. All right. Now our object normally gets placed on a glass slide. All right. So we'll be using this tomorrow. Ideally, I'm going to get a bunch of little letter E's, and you're going to put a little letter E on there, and then we're going to cover that particular specimen, because I guess instead of an object, they call it a specimen in terms of biology. What we do is we put on something called a cover slip, and a cover slip is a little piece of plastic which then you sandwich your specimen in between the slide and the cover slip to keep it from getting away and to keep it nice and flat. And once you've got your specimen in there, you put your specimen here on the, I think it's called a stage. Yeah, stage with stage clips, and you allow the glass slide to slide under the stage clips, and that kind of holds that glass slide in place. So you've got your object, and this, if we were talking in terms of our optics, you've got your object, which is right there, and then you're going to shine some light at it. Now the light source on these are little mirrors. So what you have to do with this is you've got to kind of pick a light source, and then you got to kind of reflect the light off of the mirror up into the microscope, which can be a little tricky. So what we've done is we've purchased a bunch of these little light sources. You know those little lights that you can stick to the wall, right? If you got a little thing there, you know, you can buy the commercials. These are really cheap. So I figured out that if we have these light sources, we can place this right here. Now, if you put it straight like that, you're just about blinding yourself as you look into the microscope. So I found that if you took some paper and you covered up the light, it was still producing enough light that you could see very nicely, because it makes the light kind of go diffuse and spread out instead of it being super bright. So if you take a piece of paper and you cover that up, and we might have to use some tape here, you get some good light source kind of coming through that way, okay? All right? Now, that's an optical microscope. I'm going to go over the parts here in a moment and how it looks, and I might even show you the way it looks when you try to take a picture of it. I'll talk about that in a moment. Now, there are many other types of microscopes. The electron microscope uses, instead of light, electrons. A beam of electrons from an electrical current is shot at a specimen and bounces off and reflects. And then sensors catch those electrons, and the way they bounce can be worked backwards to look at the structure of what you're looking at. You can kind of the computer can do some mathematical work backwards. Now, why is an electron microscope better than a light microscope? an electron microscope can look at much smaller objects than a light microscope. You've probably seen electron microscope diagrams before. If you've ever seen those images of like a fly's eye or a fly's leg where you've got all these furry things hanging from it. Have you ever seen that before? No? You've never seen electron microscope pictures? That's from an electron microscope. Oh, here, let's check here, you guys. Okay, what do we got? Let's go to here, and let's get rid of this, and let's do this, and let's go with scanning, electron, well, let's just go scanning electron microscope, and let's do images. All right, and so that's what the scanning electron microscope looks like. Specimens. Those aren't good ones. That's terrible. Scanning electron microscope images. That's not bad. That's not a bad one there. You can see the little cell kind of, that's pollen. All right, that's not a bad one. So that's the detail seen in different types of pollen. And you can't see that detail, right? And this is three-dimensional detail. So these are scanning electron microscope images. See if we got any others. Oh, that's a nice, good, disgusting one. That's two types of bacteria, I would think. Oh, here's an interesting structure. I have no idea what that structure is. There you can see it's got the magnification in the bottom. That little line represents 200 micrometers. Phenomenally small, little tiny pieces. Again, this looks like something growing on a surface. That looks like an artificially made surface, and something's kind of growing on it. That's kind of cool. Okay. So a scanning electron microscope allows you to see in quite detail. Confocal microscope, I don't know how those work. There's all kinds of different microscopes. I worked with something called a scanning tunneling microscope that allows you to look at almost atomic sizes. And this is looking at much larger sizes, the size of a cell. We're talking about 1 micrometer that we're looking at here, where that one was like even smaller micrometers, and then we can get into even smaller there. Okay, so there is the microscope itself, and I was going to get you to use this to label your picture. So if you've got your picture, you can kind of try to figure out which piece is which. If you don't have the picture, I suppose you could take a picture of that. Sure you could. Like, that would be fair. There'd be nothing wrong with that. I am going to make you label a diagram of a microscope at some point, all right? So you could do that. But if you could label it, that would be great. Yeah, but taking a picture would be fine as well. Is that better? Does that make a better scenario? Let's just talk about what these pieces are and what they do. Okay? All right, starting with the, well, actually, you know what I'm going to do? I'm going to start with where the specimen is. The specimen is down here on the stage, all right? And below that, we've got the light source. This says the illuminator, but it's a light source, essentially. And as the light travels up through the specimen, it then continues on, and you end up with what is called the objective lens. The objective lens is closest to the object, hence it's called the objective lens. And you'll notice the objective lens is connected to this piece, and it actually rotates around, all right? So I can rotate through these various things. So there are 3 objective lenses on a classic student microscope, and they have different magnifications. We'll talk about that in a moment. The smallest one being the lowest, and the longest one having the highest magnification. And I can choose the magnification depending on how I rotate this thing. And this is called, the thing that rotates is called the revolving nose piece. Funny name. I don't know why. I guess it looks like a nose a little bit. But it's called the revolving nose piece because it can rotate around and it can change the objective lenses. As you move up, you get the tube. which separates the first lens from the second lens, and that separates the two lenses so that they can create a nice focused image. If you brought them too close to each other, you wouldn't get a focused image. This second lens, and this one I can remove it from because there's a little screw missing, but this second lens is called the eyepiece. And there you can see it there, and that's because it's closest to your eye. So the light comes through the objective lens and then up to the eyepiece, all right? Now that's the main body of the microscope that does the kind of magnification. So how do you get something in focus? Well, first you always start on the low power objective. So you always begin at the lowest power. And when you focus, you have to move this tube up and down. And under low power, you use the larger knob, which is called the coarse focus. Coarse meaning it moves a lot. In other words, doesn't move a tiny little amount. The word we use to describe the tiny little movement is a fine focus, the last fine tuning of the focus. So when you focus, you adjust the coarse adjustment under low power. And once you get a nice clear image under low power, you then change to medium power. And when you focus under medium and high power, you only turn the fine adjustment knob, because the coarse adjustment knob gets you to where you need to be. And then when you go to medium power, you just have to move it a little bit in order to get it into focus. If you turn the big knob, you're going to lose it out of focus, and the fine adjustment knob is not going to get it into focus. So you got to go back to low, get it with the coarse. So in order to focus this, we just move the fine adjustment knob, and then that gets nicely into focus, and then you can go to high power, and you can then adjust again with the fine, and again you see more and more magnification as you move up in power, all right? Now in a moment I'm going to show you the way I plan on maybe taking a picture of this, and that's going to save us having to do lots of drawings. That's the way I want to kind of make this work, all right? You should never ever adjust the coarse adjustment knob under high focus, because I don't know if you noticed that the lens is now very close to the slide, and the slide is glass. So if you crank that coarse adjustment knob, which moves it a long way, you can break the slide because you can jam the lens up against the slide. So always only fine for medium and high, coarse for the low. That's where you start, all right? You start at low, focus, go to medium, focus, go to high, okay? Sometimes in our labs, I'm going to ask you to take a picture under low, go to medium, take a picture under medium, and then go to high, and then we can compare those pictures. Some other important things that go to this little screw here is called a rack stop, and I don't usually normally have you worry about it. I don't even think this one has it on here. It does have a little stop there. That's supposed to prevent that lens from crashing into the glass. So if you brought it down too low, it should stop it. But sometimes they're adjusted, and sometimes they're not in the right spots. So they never choose move that coarse under the big lens, or you'll break it. So that's not one that I usually worry about for labeling. The arm, yes. When you transport a microscope, all right, let's say we have it on a cart and you're going to get it to your workstation, you always grab it by the arm with one hand, and we ask you to put your other hand underneath it as you're transporting it, two hands, and then when you get to your workstation, you can put it down, all right? And that's because we don't want you to drop these things. They're about $350 a piece, okay? And they're made up of tiny little pieces. If you drop it, it's no good. It will break, okay? I know on that picture that you have on your diagram, it points to this location here, I believe. I don't really have a name for it. You could call it a pivot, but there's nothing special about it. All that pivot does is it allows you to move the microscope back a little bit, so that if you're not quite as tall and you want to work in this level, and if you're a little taller, you can work down like this, all right? So it just allows you to pivot the microscope a little bit. It's not a safety thing in any way. And then obviously at the very bottom, you have what is called the base, okay? So if you've got the labeled picture, I think you can find all the parts, right, on your little 2D picture. If you don't, this is online. You can always go back because we are going to have some labeling on, on a test or a quiz. So you can always go back to my PowerPoint on that day. You can maybe make a note right now, and you can look at that. All right. Now, at this point, I want to talk a little bit about magnification. So you can write this down. Now, we've talked about magnification already, and we had a magnification equation to calculate the magnification of a lens, which depended on where the object was and where the image was, right? Remember we had those nice equations, magnification, negative di over dou or high over ho, right? Now, due to the construction of the microscope, the object is always going to be a particular distance away from the objective lens, which means we can pre-calculate the magnification because whoever built the microscope has already done the math for you. They've taken the die, they've taken the dough, they've done their work, and they get a particular magnification. And that magnification is written on each of the objective lens. The smallest one is typically 10. Sorry, wait a minute, 4. Take a look here. 4. The smallest one is typically 4. The medium guy is typically 10. And the big guy is typically 40. I'm going to take a look there. Yeah. So the smallest one is typically 4, 10, 40. 4, 10, 40. I don't use these very often. You can have different ones. Depends on what the manufacturer builds it to be. But typically ours are 4, 10, and 40. Now, that's already built in the negative die and the dough and all that stuff. So we don't have to go back and do the optics math. We know what the magnification of these are. They're labeled right on it. Now, the eyepiece also has a magnification, all right? In actual fact, the objective lens makes a real image, and then the eyepiece simply magnifies that image like a magnifying glass. So it creates a virtual image, which is why we have to look in this thing to see the image. We're not projecting it on the ceiling. And again, because of the construction of the microscope, that there's a specific distance between the objective lens and the eyepiece, that the creators have determined exactly what the magnification is, and there it is right there. And for most of these, it's times 10. So this magnifies by 10, and this magnifies by either 4, 10, or 40. When you have two lens systems working together, the overall magnification is a multiplication of the two magnifications. So the ocular power times the objective power. Another word for eyepiece is ocular, the eye, all right? So the ocular power is the power on the eyepiece. The objective power is the power on the objective lens. And in order to get the overall magnification, you simply multiply them, all right? So if one lens makes it 4 times larger and the other lens makes it 10 times larger, then you will ultimately have a 40 times magnification. Does that make sense? It is very straightforward. Again, I'm not strong at it, and therefore I'm going nice and slow. So again, you can write that all down. And actually, as you're writing that, I'm going to shut off my recorder here, because at this point I would direct you towards the labeling page, which we've already discussed. And at the bottom of that labeling page, there's a little bit of, try to explain what does what from what I've explained to you in terms of the parts. And then there's a little magnification calculation. So in a moment, I'm going to direct you to that. And then also, I want to talk about getting into the iPads and setting up these accounts so that we can, instead of having to draw all these, we can take pictures and put them into documents and things like that. So I'm just going to shut off the recorder at this point.